Hello, everyone, students, friends, alumni. Welcome you all to join the Hong Kong U Science Public Lectures. My name is Chi Ming Chi, the head of Chemistry Department. Good afternoon. I think we have, today we have a very good speaker, Dr. Ying from our department. So to remember, it's from Chemistry Department. She is going to speak on a topic of very much current interest. I suppose everyone come across the name mRNA because of the COVID-19 pandemic, the beyond that. So everyone will be very curious about what is meant by mRNA or RNA. Actually, I'm also very curious about this. So Dr. Ying, today she will give a introduction and also the presentation. On the lecture title is RNA-based therapeutic approaches. The abstract of her lecture, actually everyone who has, okay? So maybe if you don't know about RNA, maybe you go through Dr. Ying's lectures. Eventually, you will find out. So now I pass the floor to Dr. Ying Li.
Thanks, Professor Ji, for the kind introduction. And welcome everyone to today's Hong Kong U Science Public Lecture. So today I hope to introduce you to about the RNA-based therapeutic approaches. In this lecture, we're going to go through a brief history about the drug development as well as the RNA biology. At the end, we will land on the mRNA vaccine, and hopefully you can learn a lot more from today's lectures. When it comes to therapeutic approaches, one major component is really the drugs that help us to fight against different diseases. The human civilization almost equals to the history of drug development. Our ancestors take advantage of many different materials available from the natural systems to facilitate us to fight many different diseases. And shown here are just a few examples about the active components from many different natural resources. We even apply some of these before we actually know about the active components. Although the structures are shown quite complex here, they are available from our natural systems. Some are isolated from plants, some are from bacteria or fungus, some are isolated from animals. So in the past, they have been demonstrated to have many different important therapeutic effects. And over the years, people have encountered a problem. People realize that in order to get one therapeutic drug, they actually need to start from millions and hundreds of millions compounds to start with for the initial screening. And thanks to the development of modern technology, high throughput screening in early 1990s, this approach does seems to provide a much better approach to develop a drug. But however, this high throughput screening is so modern technology, our chemical space actually didn't catch up. So in around 2019, there is a very interesting observation from the scientists in the drug development field. They realized that over the past several decades, there is a trend that interestingly saying the percentage of the drug being input into the phase 1 clinical trial, the percentage ultimately becomes the drug available for human use is higher in the biologics compared to the small molecules. So that is partially contributed by before 1996, we don't really look into the biologics. We didn't really taking advantage them to be developed into a potential drug candidate. But meanwhile, it's also pointing towards a direction saying potentially the biologics could be very useful resources for us to dig into new drug development. And what are the biologics to be exact? They are actually these biomolecules that presented in our body. Just to name the three most representative biomolecules in our body, the DNA, RNA, and the protein. They are actually connected in our body. The DNA usually is transcribed into RNA, and then RNA will be translated into protein. This is the old like central dogma. But due to the development of new technologies, people realize that this is not a single linear like information transduction. RNA can also be reverse transcribed into DNA and also can regulate the DNA transcription and also can regulate the protein folding and etc. Most important is that people realize about 93% of our DNA will be transcribed into RNA, but only less than 2% of that will actually encoded for protein. So this is the racial impression for you to appreciate that there are a lot more RNA. We don't really know what's their biological functions. And utilizing these like biomolecules for potential drug development, they come with their own like advantage and a disadvantage. Taking this DNA, for example, they have to enter the nucleus of the cells in order to take effect. And also for DNA, they also bear the like adverse effect is they potentially will disrupt human genome. So people are relatively reluctant to look into the DNA application part. And on the other side, protein, when protein are being generated, they already undergo this transcription and the translation process. So that itself already takes a lot of time. And meanwhile, protein are usually come with big size, and that will take time and effort to allow the protein to fold properly and become biologically active. And what's more, in our outside our body, the protein that we obtained, usually it's very hard to achieve the necessary post-translational modifications. And these modifications are actually crucial for these proteins to become biologically active. So with these, the RNA format would actually have its unique advantage. The RNA can take effect in the cytosol, meaning that they don't need to cross the nuclear membrane in order to execute its biological functions. And what's more, the RNA can degrade rather rapidly. So keep this in mind, RNA can degrade rather rapidly. This actually is the double-edged sword. When you take it into the maximum like advantage, it means the RNA will not interfere with your body in the long term, and they will be degrade and get rid of our body like within a short period of time. But meanwhile, you also need to fight against the degradation property of the RNA to ensure they can carry out the biological functions before they disappear from our body. So before we really look into the RNA therapeutics, let's take a brief look about the RNA biology history. So back in 1930s, people already realized that the RNA and the DNA have very distinct chemical properties. And shown on the picture here, both DNA and RNA, they have the three base as the common similarity. And these bases are shown as these sticks on this line here. And for DNA, most of the time, they adopt this double helix structure. And there is this base pairing between the different bases are contributing to this double helix structure. And because of the double helix structure, DNA molecules are considered to be rigid. On a different scenario, as the RNA, they have a different ribose chemical structure. They have a very reactive 2' hydroxy group here. And also they have a different base uracil compared to the thymidine in the DNA. because of this different like structural chemical properties, RNA usually adopt this linear strand conformation. And bear in mind that this linear strand conformation is also very important for RNA therapeutics. Because it's single-stranded, it's very flexible. So the RNA can adopt many different conformations, and that conformations later on turn out to be very important for their biological functions in terms of a therapeutic approach. And 20 years after the discovery of RNA and the DNA chemistry part, the three major RNA has been discovered in the following 15 years, including the messenger RNA, which has unique structure features, including this poly A tail at the end, which will regulate its biodegradability, as well as a 5' camp that will tell the ribosome RNA to come to complex with this messenger RNA and initiate the protein translation. And the tRNA is the one that connected many different amino acids at the one end that will using the messenger RNA as the template to allow the sequential adding amino acids to the growing peptide chain, and eventually will lead to the protein synthesis. From this history of RNA biology, I really would like to point out this one. RNA polymerase is purified and discovered also around the early stage of RNA biology history. The development of RNA polymerase allowed the scientists to get the access to many different RNAs based on the DNA template. So they can simply decide many different DNA sequence and then use the RNA polymerase to use the DNA as a template to achieve their desired RNA species. So that's the milestone in RNA biology development. Then in the following like around 10 years, many different breakthroughs happened in the RNA biology field. I also would like to point this one out. Around that time, people realized there's a specific enzyme that can transcribe the RNA back to the DNA format. So this is related to, I mentioned, RNA is not stable. It can degrade rather rapidly. So in the past, when people are working with RNA, the RNA are also happily degrading at the same time. So it's very difficult to work with RNA in the past. But this reverse transcriptase allows people to lock the information from the RNA back to the DNA and then conducted the sequencing experiment to really know about what's the different sequences of RNA they have been working with. So this is another milestone that ultimately will lead to the RNA therapeutic part. The next few decades, all the way maybe fast forward to early 2000, there's one particular kind of RNA it was significantly studied and developed, and this is the small RNA. And this small RNA was identified to be very ubiquitously generated, and also they have been confirmed to execute their gene regulation functions by post-transcriptional gene silencing method. So let's take a look at these small RNAs. They are also generated from our DNA, and some of the pathway contributing to these microRNA, those short RNA, usually about 20 nucleotides, are from a structure of RNA very similar to the messenger RNA I mentioned earlier. It also has the 5' cap and also the poly A tails. But because of the special hairpin structure adopted in the middle, these type of microRNA can interact with the certain protein machinery in our body in the nucleus. And then the outcome of this interaction will provide this shorter hairpin structure. And this shorter helping pre-microRNA will eventually be transported outside the nucleus and enter the cytosol. in the cytosol, this helping RNA will encounter a different protein, and this protein named dicer will cleave off the loop region of this microRNA, and eventually only one strand of this double-strand RNA will be kept and form an active complex with the protein machinery in the cytosol. This small strand of the microRNA is available to base pairing with the target mRNA, and that targeting part, once happened, will regulate the mRNA lifetime as well as its biological functions. So indeed, we are learning from the natural mechanism of these microRNA to develop these RNA therapeutics. And this part of base pairing process happens to the majority of the RNA therapeutics developed so far. So RNA therapeutics itself does come with many different flavors. Mostly can be divided into 4 categories. One part is related to RNA aptamer. It's more talking about the binding towards protein partners. And the major fraction of this antisense oligo, as well as the RNAi, they are all utilizing the knowledge we learned from small RNA, the microRNA. And we can appreciate that the mRNA shown as the blue strand here, they are the target. And the yellow strand shown here are the therapeutic RNA components. Because of the base pairing, they will interact with their designated mRNA, and then this binding will change the biological functions of this mRNA. Sometimes the binding will lead to the degradation of the RNA. Sometimes the binding will lead to a different format of the spliced RNA to be generated. Either one of them are leading towards the different outcome of the protein. So if the mRNA is being degraded, then there is no protein going to be generated. But if the mRNA is being spliced into a different format, then a different protein can be generated. And these are ultimately lead to the promote degradation or inhibition of the translation for these therapeutic RNA to take their functions. A different mechanism are complied by mRNA. They are utilizing the sequence of the RNA to instruct the cells to make a protein that is carried out by this mRNA sequence. And this newly generated protein will also be functional to regulate the cell fate or cell status. And one of the proteins that's being generated are named as antigen. These are also one type of protein, but specifically refer to the protein that's being displayed at the cellular membrane. And those protein at the cellular membrane will teach our body, especially the immune cells, to develop the immune systems. And that is partially contributed to the mRNA vaccine that later on I will touch upon. So following this, I would really like to focus our attention on 2 different therapeutics that has been approved in the past decade. One of them is this RNA aptamer. RNA aptamer is really a small oligoRNA that's useful to regulate the binding towards the proteins. And it has been utilized in combat a disease called macular degeneration. This disease is also named as age-related macular degeneration, short as AMD. So in normal vision, the people will look at the world with very bright color and clear pictures. However, from the same view from the patient with AMD, the vision will significantly be degraded. Usually come with the vision lost in the center of the view, and also the color and sensation become very blurred. And this blurred picture are caused by the formation of new blood vessels and also the increased permeability from these leaky blood vessels. So after digging into the disease mechanism of this age-related macular degeneration, people realize that it's because of the overexpression of one particular vesicular endothelial growth factor that's presented at the cellular membrane, functioning as a signal protein. This overexpression of VEGR lead to the formation of leaky blood vessels and eventually contributed to the vision loss and degradation. So if the logic will flow as if we can successfully perturb the function of this VEGR, prevent it from being conduct the signaling transduction, then we will be able to inhibit the leaky blood vessel to be generated and restore the vision of the patient. So indeed, in 2014, this RNA aptamer-based therapeutics has been approved by FDA. And this particular RNA aptamer is composed of 27 nucleotide in the active core structure, and coupled with PEG chains at one end, and also another modifications to ensure the RNA aptamer being biologically stable. So at the core of this RNA aptamer, out of the 27 nucleotide, only 2 nucleotides shown as this light blue are actually the normal natural nucleotide. All the others are undergoing many different modifications, such as methylation at the 2' hydroxy group site, or methylation at the cytidine site. So these modifications are providing the opportunities to first improving the biodegradability, and second, allowing the RNA aptamer to adopt even stronger binding affinity towards the target protein, this VEGR, responsible for the formation of leaky blood vessels. So if you take a look at the specific structure, this PEG chain really is composing of 2 polyethylene polymer chains. And these PEG polymer chains are very important to prevent this RNA aptamer therapeutics to non-specifically bind to other proteins, and thus can providing a better selectivity towards this VEGR and improve the therapeutic effect. So although this RNA aptamer therapeutics has been approved in 2014, I have to mention that this RNA therapeutics has already been discontinued, because shortly after the report of this RNA aptamer, people realized that the antibody approach also can provide similar biological functions. But nevertheless, this 2014 is one of the first RNA-based therapeutics that is approved by FDA. So does initiate a field that can utilize RNA as the therapeutic approach to combat disease. Now let's dig a little bit into the severe acute respiratory syndrome called Neurovirus 2, SARS-CoV-2. So hopefully every one of us are already familiar with this picture shown here. This is depicting a viral particle, which is the major component causing the pandemic ever since 2019. And this viral particle are typically about 60 to 140 nanometers. And outer layer of these viral particles, these red spikes proteins are the spike proteins that's responsible for interacting with the human cells at the very first stage. And inside these viral particles, there are RNA informations embedded. And these are the gene information of the viral particle, and will contribute to the virus replication in our human bodies and cause disease afterwards. So shortly after the announcement of this pandemic outbreak at the end of 2019, almost within 1 year, the world has been able to provide this mRNA vaccine as a therapeutic approach to combat this devastating disease. This is really a very short period of time, comparing with the old approaches for drug development, which typically takes about 10 years. Also considered very short when you compare it with the old method to generate vaccines to combat disease, which usually will take about 3 to 4 years. So what is the basic principle about these mRNA, why they are actually effective to help us combat the disease? So really the mRNA vaccine, when it's injected into your body, it will enter some of your cells in your body. And this mRNA entity will firstly be encompassed by the lipid in the cellular membrane. And after a few times, the mRNA vaccine will eventually break through these lipid vesicles. And then at this stage, the cellular machinery inside the cells will treat the mRNA vaccine as a normal resident inside the cells and happily translate the mRNA vaccine into the designed protein, which is responsible for the therapeutic effect. So the protein that is being generated after the mRNA vaccine has been successfully translated inside the cells, these proteins will eventually be presented at the cellular membrane to teach the immune cells to combat with this particular antigen. And this approach really encompasses of 2 key components. One is this lipid nanoparticle, and the other is the mRNA vaccine. So this is talking about when you are designing the mRNA vaccine, what kind of mRNA sequence would you like to incorporate? Another part is this lipid nanoparticle. Since mRNA can degrade rather rapidly, how we can ensure that this mRNA can be protected very well and help them to enter our cellular body and then execute their biological functions. So in terms of the mRNA vaccine sequence, again, we are learning from the nature. So this is the cartoon of this virus particle we've seen in the previous slides. Really, it's the spike protein that's interacting with the receptor on the cellular membrane of our cells, then allowing this virus particle to be uptake by our cells and then cause the infection and the disease. So shortly after the releasing of the COVID-19 genomic sequence, people realized that this yellow segment is really corresponding to the generation of the spike protein on the viral particle surface. So taking advantage of this segment of the RNA sequence, scientists have been engineered that sequence very extensively to allow the sequence can be successfully and efficiently being translated in our cells. So many different modifications have been adopted onto this particular mRNA sequence. Some of them are involved with nucleoside modification. Some of them are working on the GC content of the sequence ratio. Some of that is working on the cap structure, trying to teach the cellular machineries to well recognize this particular mRNA sequence and initiate the protein translation part. Also engineer the end structure of this mRNA sequence, so this mRNA can survive for a certain period of time after entering the cellular environment before being degraded. So allowing enough time frame, allowing this mRNA sequence to be successfully converted into the protein product. So here is when the mRNA vaccine is being uptake by our cells. Once it's being able to escape from this endosome, this mRNA vaccine will encounter the cellular machinery to generate this protein product. The engineering of the mRNA vaccine will also introduce 2 mutations so that the protein product can stably adopt this S protein trimer conformation. So this type of S protein trimer can locate at the cellular membrane for a longer period of time, and that will lead to a better immune response generated by our body to protect ourselves from the viral particle infections. And these 2 are really the key component of the mRNA vaccine part. So another technique very important I mentioned earlier is utilizing the lipid nanoparticles to protect the mRNA vaccine and also help the mRNA vaccine to enter the cells. And this is related to the fact that the RNA can degrade rather rapidly. So indeed, the technology needs to be developed to counter this rapid degradation of the RNA. So 2 different mRNA vaccine, one is from Moderna, the other is from BioNTech, Pfizer. Both of them adopted this lipid nanoparticle approaches to facilitate the delivery of mRNA vaccine. They enjoy the similar component shown here in the middle as the helper lipid, such as the normal DSPC or the cholesterol. Hopefully most of you are familiar with. It's presented in our body. It's also a very important component to regulate the cellular membrane fluidicity. But different, other than these helper lipids, 2 different companies actually turn to 2 different ionizable cationic lipid, as well as the PEG lipid for the real lipid nanoparticle formation. But they do enjoy the similarity between them. So for example, in terms of this ionizable cationic lipid, both of them have this nitrogen atom in the middle. And this nitrogen atom is very important to allow the pH sensitivity inside the cell, inside the cells. This nitrogen will later on be protonated at the lower pH environment, and this protonated lipid will disrupt the lipid nanoparticle and allowing the mRNA vaccine to be released from the endosome and encounter the cellular machinery to carry out the translation process. And also the PIC conjugated lipid, their structure are also slightly different, but they still will encounter this like bilayer of the bitail of the lipid tail, as well as this polyethylene glycol chains that again will provide this protecting effect. So this lipid nanoparticle will not non-specifically interact with other non-specific proteins inside the cells. So these will also contribute to the stability of the lipid nanoparticle formation. So with that, with the mRNA vaccine sequence being identified, with the lipid nanoparticle components being identified, the current technology of this flow technology will mix these 2 components together in a regular manner so that they can mix together to generate the ultimate mRNA vaccine containing lipid nanoparticles. And this lipid nanoparticles is the part will enter the cell environment and to initiate this mRNA being translated into protein, presented into cellular membrane, and initiate the whole process of immune response generation. And when you compare the mRNA vaccine generation history to the whole history of mRNA vaccine development, you would appreciate that 1 year is really a considerably short period of time. And the reason we can actually, or the world can actually achieve this RNA vaccine within 1 year, thanks to the previous 3 decades of effort. So the whole process really initiates even before 1990s until recently, that within 1 year, the world will be able to come up with this mRNA vaccine to combat the COVID-19. And what I really want to point out 2 milestones in this timeline of mRNA vaccine before COVID period. One of that, it happens shortly after the 1990s. This is the first concept of utilizing mRNA to regulate the another mRNA content inside the cells. So at that time, the scientists realized that when the mice originally being infected by influenza virus, when injecting a certain mRNA species, this influenza virus content can be reduced. So that is the first proof of concept that by injecting a particular RNA species into a mammalian cell, they can regulate the level of another RNA content inside the cells. So this is the like initial point of this mRNA vaccine development. However, another milestone was not achieved until almost 15 years later. This is when the scientists at Duke University realized that the toxicity of the mRNA vaccine can finally be eliminated or suppressed to the minimum level. They realized that by doing extensive nucleoside modification on the mRNA species, they will be able to achieve the similar therapeutic effect without introducing the adverse immune response caused by injecting mRNA directly into the cells. So these 2 are the 2 very important milestones marked in the development of mRNA vaccines. Without this development from the beginning, as well as eliminating the cytotoxicity effect from the mRNA species, there is no way the world will be able to achieve the mRNA vaccine just within 1 year of detection. So here is trying to convince everyone the mRNA vaccine development really is a collaborative work around the world, around many different pharmaceutical companies, among many different academic institutes. Here is the network analysis of this COVID-19 mRNA vaccine patterns. So this is talking about in order allowing this mRNA vaccine technology to mature over the years, many different patents have been filed, and each one of them are very indispensable to the final success of this mRNA vaccine to be generated and successfully applied in human beings. To name a few, of course, we know Moderna and BioNTech, the 2 companies that generated the first 2 mRNA vaccine for human utilization. And also, not to mention, another company are also heavily working towards the RNA vaccine part, and also many different universities such as UPenn, as well as University of British Columbia. Many other companies have joined this journey. Even Tesla are contributing to this patent network of COVID-19 mRNA vaccine. And here I hope to give you a future view about the mRNA vaccine or RNA-based therapeutics into the future. Here listed are some of them has already been approved by FDA. Many of them are related to the antisense oligo I mentioned earlier. They rely on this RNA interactions to execute their biological functions. And other than these antisense oligo or siRNA, there are many other options related to this mRNA therapeutics are currently available in the phase 1, phase 2, phase 3 trials. And these RNA-based therapeutics, they are actually touching upon many different fields, many different disease, including cancer, but also including other like cardiovascular disease or other neurological disease. So indeed, the generation of mRNA vaccine for COVID-19 is really marking the starting point for this RNA vaccine to be like invaluable to human civilization. This is the starting point for mRNA vaccine will ultimately become the major component going to fight many different diseases in the field. So with that, I would like to end today's lecture here and invite any questions from the audience. You are welcome to type into the chat or maybe raise your hand or voice yourself out. Okay, there is one question. Good afternoon, Professor. Do you think RNA can be used to manufacture influenza vaccine instead of using inactive virus? Is it better? I think people are really looking into that directions. But one part very important to bear in mind is the influenza virus is very hard to combat because they are mutate themselves very rapidly and unpredictably. So I would believe that the mRNA vaccine can contribute to the fact that it will become easier and easier to generate the vaccine to fight against influenza. But in terms of whether you can predict the influenza mutation better, that's completely different stories. There's another question talking about would the modified mRNA be reversely transcribed back into DNA and thus contaminate the DNA with deformed DNA there? Very good question. So indeed, it's you highlighted the importance of these RNA modifications. One very important part is the enzymes in our body responsible for reverse transcribe the RNA back to DNA. They are very specific. They won't be able to tolerate these modifications. So in other words, these modified mRNA are considered relatively safe. They won't be able to transcribe into DNA and then mess up with the human genomes. And also at the same time, these modified mRNA, although their degradability is suppressed, but they are still prone to degradation. So their survival time inside the cells are still limited, also prevented them to be successfully transcribed into the DNA format. Another question related to, may I know more about how RNA can be applied on controlling cancer cells to spread in humans? Very good questions. And this is still ongoing development. And one of the direction is talking about how human can sense the cancer cell development progression and how human can suppress the cancer cell survival. usually most of the time when people are detecting cancer cells, it's usually happen at the late stage. So at that time, people already doesn't have much more therapeutics to control that. This is meaning the human sensation of the existence of cancer are relatively late. So this RNA-based vaccine to treat cancer cells is trying to bring forward our sensation of the existence of cancer cells. So when cancer cells are generated, they will also produce certain informations into the environment. How we can detect those information in an earlier stage, then these potential mRNA vaccine can teach our body to pick up those information much earlier than nowadays therapeutic approach. In that sense, these mRNA vaccine can control the cancer cells to spread in the human bodies. Just to verify, will you share the PPT after the session? I think it will, I'm totally okay with share the PPT. The format of how this PPT will be shared will subjected to the Faculty of Science decision. Can the mRNA being translated to vaccine protein in vitro? Yes, that's possible. And this is considered, but if you are translated the mRNA into the protein in vitro, then you will face the problem as how you would apply this vaccine protein into the body. So I also mentioned in the talk that the protein needs to take time and effort to fold properly. Only when they are folded into the correct conformation, they are biologically active. And also protein itself is also considered not stable in terms of when their folding is disrupted, they will lose their activity. So how you can provide this active format of protein inside the cells to carry out its biological functions, that's also unmet challenging. So that's why mRNA vaccine at this point is considered to be an advantage. It will allow the cell utilizing its own resources to generate the protein and utilizing the own resources to present the generated protein to the cellular membrane and then interact with the immune cells to initiate the immune response. Hi, good afternoon. This is Ying from Malaysia. As we know, mutations occurs very rapidly. Can the mRNA technology be able to compete with that speed? Thank you. Yes, so this is a question actually can be answered at this stage by the mRNA vaccine that's already been generated. So the COVID-19 virus has already mutated from the alpha format, beta, delta, and now Omicron. But actually the mRNA vaccine generated, the first two are still being able to protect us from the mutations of this virus. The reason is these mutations does not really change the infection pattern of these virus particles. They are still relying on the spike protein to interact with our cellular membrane to initiate the infection process. So the mRNA vaccine design based on the spike protein are still being able to fight against the mutated virus and protect us from these mutations. And in the future, and also currently, there are research groups trying to develop even better mRNA technology to further protect us from mutation, meaning bring back the fighting, preventing infection ability of the RNA to even higher level. The recapped video, another question from here, would RNAi gene silencing be more leaky than silencing by transcriptional factors? this is actually composing many different components. So first of all, how to define the leakiness, right? Another is what's the definition of leakiness of transcriptional factors. So transcriptional factors, according to my personal understanding, they are still peptide level or protein level. So their silencing effect will based on protein RNA or protein DNA interactions. And the RNAi gene silencing part will rely on the base pairing interactions between the small RNA sequence versus the corresponding sequence in the mRNA level. So their mechanism is different. And also I would like to point it out that the RNA sequence can allow for many different modifications to enhance the binding affinity. So this is making this RNA silencing effect to be much more better regulated and improved compared to the transcriptional factors aspect. Another question here is asking whether RNA-based therapeutics are viable considering personalized medicine. Yes, exactly. So this is again coupled with how this RNA therapeutics can be designed. Most of the current disease, they are related to this inherited disease, and also different person have different mutations in their genome system. So this RNA vaccine, because this RNA-based therapeutics, because the sequence of the RNA can be very well exchanged or regulated or modified, that they have a better opportunity to be tuned towards each individual patient in order to carry out the maximum therapeutic effect. What endosome is and its function? Is it responsible for releasing substance in the particles? Okay, due to the time limit, I didn't really mention about that. So let's take example, take a look at here. So this is the, and this is the part introducing when the mRNA vaccine lipid nanoparticle, this outer layer is still the lipid. When this lipid nanoparticle encounters the cellular membrane, there is lipid-lipid interaction happenings. So in order for this lipid nanoparticle to enter the cell, actually some of the cellular membrane are contributed, wrapping around the mRNA lipid nanoparticles. So these are the endosome I was talking about. So once these like endosome is accumulated for a certain period of time inside the cells, their pH value will be changed because these endosome entity will also interact with other entities inside the cells, such as lysosome. And lysosome is known to have lower pH, and that lower pH eventually will trigger the release of the mRNA vaccine into the cellular environment. Can the mRNA introduced encode more conserved regions of the spike antigen, so the vaccine evading possibility of the CO2 variants may be lower? You are exactly on the right track. I think I have heard that the current like research is exactly looking towards that. People are also predicting what other mutation patterns would uptake by the spike antigens, and they are designing the mRNA corresponding to that mutation directions and trying to produce the RNA vaccine that can combine all kinds of mutated virus. Is it said that mRNA can only go out of the nucleus membrane, but not the other way around? However, would reversely transcribed DNAs re-enter the nucleus membrane? So first of all, this is more related to the interactions between the RNA and the protein. Indeed, sometimes the RNA can be shoveled back into the nucleus, but many of that happened at a like controlled manner. And also another aspect of that is related to the RNA lifetime. Many of those transported backed RNA, they are specialized structure and conformation. So they have the opportunity to survive long enough to be transported back. But the mRNA design, they are fine balancing between their therapeutic effect and their degradability. So less likely these mRNA will be transported into the nucleus and then being reverse transcribed into the DNA and mess up with the genome, human genome system. Can you explain how vaccine, very important questions. I didn't mention about that. Yes, we heard about a lot that the mRNA vaccine, although it is very efficient in combating the disease, but meanwhile, almost every one of us or every person injected by the mRNA vaccine, they are having the side effect of immune response. This is talking about when you are during the injection, these RNA species still can leak a little bit into your body, and that leaked RNA will induce the immune response, and that immune response is considered as the side effect. And if you are referring to a broader aspect in terms of the side effect, and I believe that will be a case-to-case study, different patient or different people, they have different like health status, and that significantly impact on what kind of side effect can be caused by the vaccine. Is it possible to tune mRNA vaccine into drugs which allow us to swallow? You are definitely on the right track. I have read about reports around that part. Indeed, right now the mRNA vaccine, they require this cold temperature for storage and transportation is significantly limiting this vaccine to be widely applied. And also the cost is significantly. So in the right now, people are trying to develop approaches so that these mRNA vaccine can be stored at room temperature, but whether it can be converted into pills that still undergo along like developing stages, because you need to think about when you are swallowing, meaning the mRNA will enter your stomach, and your stomach is particularly low pH, and there are many other enzymes present in your stomach trying to destroy or digest the species from outside, right? So this will contributing, unlikely the mRNA pills will be developed in a short period of time. Are there any major difference between the efficacy of BioNTech and the Moderna vaccines? Which one is more protective and safe? I would believe both of them are quite protective and safe. If you really want to dig into the details, I remember, at least according to the communication with my friends, Moderna's vaccine seems to induce even larger immune response in terms of the side effect. This could be related to the PEG chain like utilized from different companies. It is saying, not confirmed yet, the Moderna vaccine utilized a longer lipid chain in their lipid nanoparticle formation, and that longer lipid nano PEG chain is contributed to the stronger immune response. Various COVID variants had spike protein mutations. how is that the M vaccine is still working? Future, would the mRNA vaccine be more robust in response to the mutations? I think I have answered the similar question in the past. So although the mutation indeed happened, but it's not happening to the extent that completely changed the infection pattern. As a result, the mRNA vaccine currently can still produce the enough like protecting agent to prevent the spike protein to interact with the cellular membrane. How likely will miRNA inhibit non-targeted mRNA sequence? Hold on. So mRNA, it does need to interact with their target mRNA in order to execute the therapeutic effect. So if it's a non-targeted mRNA, less likely they will perform this very well binding interactions. And without the binding interactions, no therapeutic effect will encounter. Oh, okay. I see. Yes. There is this. It's already about 6 p.m. There are, I think, a lot more questions are still flushing in. I will not too many. There are only 3 left, so I will quickly go through that. Will there be the chance that the modified mRNA for vaccine mutators in vivo and cause adverse effect to human beings? For vaccine mutators in vivo. So you are talking about the mRNA vaccine is being mutated inside the cells. Again, I think the mutation of the mRNA vaccine less likely will happen. First of all, there is not too many enzymes inside the cells to actually working on that. Remember, the RNA vaccine is mostly being modified significantly. So they are currently only interacting with the ribosome to translate the mRNA into proteins. Another aspect, as I also mentioned earlier, this mRNA vaccine still has a limited life window. So less likely they will survive long enough to experience the mutations inside the cells. How can the lipid nanoparticle be made so that only specific cell types uptake the mRNA inside? This is especially important for treating the cancers. So indeed, similar research are being conducted to introduce this targeting effect to the lipid nanoparticles. You can imagine it as instead of only conjugating PEG chains, you can swap that into a particular like peptide or guiding sequence to allow the lipid nanoparticle to be only effectively uptake when certain receptors are presented at the cellular membrane. And that will allow the lipid nanoparticle to be only uptake by specific cell types. Why mRNA causes more harm, such as rather than adults? I actually didn't read more on that aspect, but I believe it could be related to the different immune system development. So like young people, especially within under 20 years old, they are themselves like growing rapidly. So their metabolism as well as the immune response is different from adults like older than 20 years old. So that will contribute to why for certain like young people, they develop like even more immune response in terms of the mRNA vaccine. All right, let's come to the last question. Is it true to say that a virus will become weaker and less harm if it continues to mutate to form new variants? Unfortunately, it is not true. Scientifically speaking, the mutation of the virus should be random, and it is totally up to the statistic effect whether the mutation of the virus will generate even stronger mutant to impact the human society. All right. So looks like we have answered many questions, and hopefully we are all interested in RNA-based therapeutics, and welcome many of you will continue support or look into this direction, or maybe even join the society or field and contributed to the RNA-based therapeutics in the future. And with that, I will conclude today's lecture, and thank you everyone for your attention and questions.